let us clap for Sue as she joins us to take us on the next phase. Welcome, Sue.
Thank you, James. And hello, everybody who's already in class. We know those that are really focused on this learning, they get to class first. And everybody else dribbles in and then they miss a little bit here and there. But they can always go back and pick up stuff on Canvas. Just a recap of what we did yesterday. So yesterday we looked at the situation of Kenyan soils, and our homework was to have a look at our soils, to go out and do some tests. I don't know who has done the tests. I don't know why my camera just went off then, Tech. I don't know who has tests already, but hopefully we'll start seeing some of those coming in the class. Lots of fun. Remember there was the water infiltration test? What does that tell us? It tells us what's going to happen to our soil when it rains. We want our soils to behave like sponges, especially in today's day and age with climate change and with the issues that we're having heavier rainfall, we're having longer droughts. We want our soil to soak up as much water as possible. The difference between my farm and your farm, and we can be sharing a fence, can be colossal. It's all about how we've each looked after our soils. So if the entire area might look very dry, but if you're looking after your soil in your particular place, you're going to find that your soil will act like a sponge and you'll have a longer growing period, as well as your plants will be more resilient to the vagaries of climate change, and your soil will move with all of the work that you might have put into it. So that was the water infiltration test. Then we did the slaking test to look at whether the biology is in there and holding the soil particles together and stopping the soil from just breaking down and disintegrating. And then finally, we've looked at rhizosheath and rhizosphere and those tests, and those are showing us what is actually happening in the root zone. And as I said yesterday, and I'll re-emphasize today, oftentimes when we have a problem with our plants, with our crops, we look at the top of the plants. We measure things in terms of yields. So we wait for the yield in order to say, how did we do? But the how did we do is happening while those plants are growing. And we can correct problems that we might have. We can correct issues that we might have along the way. And we can measure them by doing some of these tests and looking in the root zone and seeing what's happening with our plants. Today, we're going to do two other things. We're going to learn a little bit more about the soil food web. What is the soil food web? We've described it briefly in the other classes, but today we're going to go in depth. I'm going to actually introduce you to one of the greats in this place, Dr. Elaine Ingham. She's an American microbiologist and a soil biology researcher. She's a founder of the Soil Food Web, Inc. She's known as a leader in soil microbiology and research of the Soil Food Web. Now, my experience with the Soil Food Web, for those of you that don't know, is I started training with the Soil Food Web in taking their foundational courses. They have a bunch of different courses. They have a foundation course, which is a 1-year-long course, and that basically takes you through understanding the Soil Food Web right through to your basic microscopy. What do those organisms look like under a microscope? And what do they tell us about the soil food web, about the health of the soils? That's one. The second part of their course that they offer is about it's called the Lab Tech course, where you now get trained with a mentor virtually using Zoom. But you need to have equipment, you need to have a microscope, you need to have the tools for the lab, and you basically are trained over, I think it's a period of 3 to 6 months, on how to really do those microbial assessments. And then finally, the third part is the consultancy course, which incorporates all of those, the foundation course, the lab tech course, and more. And if you manage to pass all of those and get the consultancy meet the Soil Food Web consultancy standards, then you can do any of the Soil Food Web work anywhere in the world. So actually, for some of you that are in here as young students who are wondering about your careers, it's actually a great opportunity to look at a career path in consultancy using the Soil Food Web program as a stepping stone to get you there. The courses are hard. You have to pass with 90% every step of the way, right the way through from the foundation. You can't get into your lab tech course unless you've passed with 90%, and you can't get into your consultancy course unless you've passed your lab tech course. It's tough, but it's rigorous and it's great. So I'm going to play a short video, like I say, of Dr. Elaningham. She gave this video about the Soil Food Web during a recent global summit meeting. So I've just taken that, which will play for you here. And I'm not going to do any more in describing who she is or what she does, because the video says it all. So let's go over to that video just now.
Thank you very much, everyone, for attending these meetings, the Soil Summit. Very happy to be able to bring this program to you. So no further ado, let's get going. I want to talk about the Soil Food Web for a few minutes this morning. What is it? What's comprised? Exactly what does it do? Those kinds of questions. And I always like to start out with this particular slide because it reminds me of a lot of the information that I need to put out. This was a picture that was taken by the Lexicon for Sustainability group out in Pennsylvania. And I love the fact that they put the Soil Food Web diagram overlaid of me out in that field, holding up my hands, kind of saying like, yeah, see, I can get dirty. The field always reminds me of what I need to talk about a little bit. When you're looking at this sort of field, one of the things you want to ask yourself is this soil or is this dirt? And some people are surprised that those are really truly two different things. When we're talking about soil, it has to have all of the food web organisms in it, and it needs to have some organic matter as well. When I look at the color of this field, this recently tilled field, I get a little concerned because this is not the rich, dark brown, 70% cocoa chocolate color that I want to see in a good soil. This light tan color says to me, we got some problems here. We don't have the complete food web. What's lacking? What's missing? What do we need to get back into the system, and how are you going to put that back into the system? Another thing that I look at when I'm looking at this particular field is all of these big clods. This dirt has been compacted, and then they came along and tilled it probably to get rid of that compaction, because your roots won't go down very far until when they hit that compaction layer at 4 to 6 inches. The roots aren't going to go any further. The roots of terrestrial plants are obligate aerobes. And when you're in a compacted condition, there's very little oxygen in there because the few microorganisms that were present in that material to begin with utilized all of the oxygen, so it's gone anaerobic, and your roots can't grow into it, or your roots will be harmed. They may well die because of that experience, and certainly they will grow out sideways then instead of down. If your roots are going out sideways, it's going to be trying to steal nutrients from the plant next to it. So you're going to have some problems. Those plants are going to be stressed. They're not getting the full nutrition that they require. So tells me quite a bit just looking at a field to discover those kinds of problems. So how do we fix those things? How do we rebuild structure? How do we get that organic matter back to what it needs to be? So this soil in this field is probably well or significantly lower than the minimum of 3% that you really need to have in soil in order to grow plants without requiring pesticides or inorganic fertilizers and such. I like to give a little bit of introduction about who I am. I am Dr. Elaine Ingham. I have all that lineup of alphabet soup after my name, ta-da. I started out as a soil microbiologist, and I've really turned into a soil ecologist, if you will. But of course, my focus is on the organisms that live in the soil. I'll let you read through the list here. You don't need me to do that for you. So looking at what the soil food web is, what the soil food web does, is really where I want to go today. So what is the food web? The processes in soil, and that's important to recognize that we're talking about soil. The processes in soil are done by the microorganisms for the most part. All the critically important things that are supposed to happen in soil happen because the biology is present. So just a reminder, if you want to go to our website and take a look at all of the different things that Soil Food Web School does, please go enjoy the website. Remember to go to all the way down to the bottom of the pages, because there's more interesting information. The links to that information down at the bottom of the page is not necessarily at the top, just to keep you on your toes. So the Soil Food Web, in a picture model, if you will, this is really a flow of carbon through the food web, or you can also think of it as the movement of energy from sunlight through this system, or any nutrient. You could reasonably be following where those nutrients are getting to in the biology that should be present in your ecosystems, in a community, in a watershed. So we want to understand how biology affects these things. I remember when I first came to Colorado State University to work on my PhD back in 1978, one of the first things that my major professor had me do was to go to every professor at Colorado State University that dealt with soil in any way. So to hydroponics people, to horticulturalists, to the crop scientists, to the soil science department, to the agronomy department, and talk to those professors about what it is that these organisms do in soil. Why are they there? What is their function and purpose? And I wanted to work on a project where we were going to look at how do you measure fungi and bacteria and protozoa and nematodes all in one sample, so we could get that information out to the grower as rapidly as possible. Could we get this information returned to the grower within 20 minutes? Typically back then, it took a good 2 to 6 weeks to get the information about just these few groups of organisms back to people. And that's way too long. Growers need that information right now. So what do these organisms do in the soil is a question I was asking to all of those professors at Colorado State University. And was I wise to put together a PhD program that focused on those organisms? Would there be a job for me when I got done with my PhD? And that every single person that I talked to there, they said, that's a bad idea. There is no job market. You're not going to have a job when you get done with your 4 years of the graduate program. These organisms aren't important in soil. They don't do anything to help your plant grow. They're just there. And I remember at the time thinking, what's wrong with these people? Because nature has kept these organisms alive and active on this planet for, well, take bacteria. We know there are, in the fossil record, there are bacteria that we can show that material is 4 billion years old, and it has bacteria in it from that time. And so Mother Nature has kept those bacteria around for 4 billion years. They have to be doing something, or Mother Nature would have kind of let them go and they wouldn't be present amongst us. How about fungi? 3.5 billion years that fungi have existed. Protozoa, a little bit shorter time. Nematodes as you go through the whole evolutionary system. These organisms have been around for a lot longer than human beings have been around. And nature doesn't keep things that aren't important, that aren't performing a function. So just because we as human beings have not paid attention to what these organisms do, or if they're important, doesn't mean we should take such a callous attitude for them. Because of course I went ahead and did my PhD work, starting to show what the role and function of these organisms were in the soil. Of course, I worked with a huge number of other people, David Coleman, Pat Reed, Vern Cole, all sorts of people at Colorado State as well as the University of Georgia. And then when I arrived here at Oregon State, continuing to work on these organisms and what they do. And so understanding what's going on. So sunlight energy, for example, is fixed through photosynthesis and pulling into carbon dioxide molecules into the plant material. It's going to store the energy from that sunlight in the carbon-carbon bond from those two carbons coming from carbon dioxide. The waste compound, oxygen, blown off into the atmosphere, for which we are eternally grateful. So we can breathe and we will return the CO2 for those plants to now recycle and use. Of course, you can't grow plant tissue on carbon-to-carbon bound materials. Our plants, nothing on this planet grows on carbon chains alone. So those sugars, which is what you would call them, the short chain or slightly complex sugar compounds being produced above ground, are going to be used as the transporters of the nutrients that the plant has to have in order to grow, that we have to have in order to grow. So these sugars are going to be sent down into the roots in order to pick up nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium, iron, zinc, all the nutrients that your plant requires. Is there any nutrient on the periodic table that your plant doesn't need? Well, some of them we don't need very much of, but almost everything on that chart of elements has to, in some concentration, is required by your plant. So all of those nutrients have to be picked up in the soil. So, okay, that sounds pretty good. Here are the roots. The sugars are going to be transported down into the soil, and your plants are going to pick up the nitrogen, phosphorus, sulfur, magnesium, calcium, etc. Well, but where did that nitrogen, phosphorus, sulfur, magnesium, calcium, etc., where did that come from? It didn't just like, boop, magically show up around the root systems of the plants. Where did it come from? How did it get here? And in high enough concentration that your plant can take up what it needs, tie those nutrients onto the sugars, and now we start calling those amino acids or proteins or lipopolysaccharides or any one of the other gazillion nutrients, organic compounds in your plant. So where did those nutrients come from? So you have to understand that this plant is going to put out exudates, which are basically sugars. And the kind of exudate it is meant to wake up, specifically wake up specific species of bacteria. It's going to be food released out into that root system, right around the roots, so that it will grow those bacteria. And those bacteria will get the message that what your plant requires is some iron, is some zinc. Same thing for fungi. The plant puts out exudates, which are specifically meant for the fungal hyphae, and they get that message to make the enzymes to be able to pull the nutrients off of the sand, silt, clays, rocks, pebbles, parent material, and start accumulating those nutrients, storing them in the fungal hyphae or in the bacterial bodies. So we're starting the process of converting those inorganic, soluble, not soluble forms of nutrients out in the rocks and pebbles and such into bacterial biomass, into an organic form inside the bacteria, inside the fungi. And then they store those nutrients until the plant, making certain that it's got lots and lots of bacteria around that root system, and oh yeah, protecting the roots of your plants from diseases and pests and other predators attacking those root systems and destroying the root system. So very important in protecting your plant. If we don't have the right biology in the soil, we're not going to be able to protect the plant. So the protectors, the castle wall, the knights in shining white armor protecting your root system, will prevent the disease-causing organisms from being able to germinate. No food, no space, not enough anything for those bad guys to grow, if you will. So, but bacteria and fungi, well, I haven't really kind of gotten around to explaining to you how it is that we get soluble inorganic nutrients around that root system in the form that your plant needs. How does that happen? We've got nutrients tied up in the bacteria and fungi, we're protecting the roots against diseases, but what's the next step? Well, you have to have the third trophic level groups of organisms, the predators of the fungi and the predators of the bacteria. So bacteria, just follow the lines to the protozoa. The two good guy protozoa, the aerobic ones, and remember everything down here needs to be aerobic or it's going to be killing your root system. It's going to be harming its ability to take up the nutrients. So we want the aerobic organisms present and functioning in that soil. So the amoebae and the flagellates. But there are bad guys in the protozoa, bad guys that we can use if we're looking at our soil using a microscope. We can see those ciliates, and if we're starting to get a lot of ciliates in our soil, we can know that things are not good, that this soil isn't really soil. How many of the bad guys do you have to have until you start calling it what it is? Dirt. So we need to have the good guys, we don't need the bad guys. How do you select against the bad guys? It's all about the conditions in that soil. You have to have the right conditions to grow the good guys. What are the conditions that grow the good guys? It's aerobic conditions. If you go anaerobic, you're going to be growing the bad guys. And that's why we call them bad guys, is because they let you know something very bad is starting to happen in your soil and you need to fix it now. Well, then also bacteria are eaten by bacterial feeding nematodes. Same thing, the good guys like it aerobic, the bad guys, they kind of want it reduced oxygen conditions. Fungi are eaten by protozoa, the fungal feeding nematodes, and I think I said fungal feeding protozoa, so I'm going to back up. So then the fungi are eaten by the fungal feeding nematodes, as well as the fungal feeding microarthropods. So when any of these predators eat any of their prey group, the nutrient concentration in bacteria is way higher than any other organism on this planet. Nothing can beat bacteria for the storage capacity of all the nutrients that your plant's going to need, or that you're going to need. So when these guys eat the bacteria, there's way too much nitrogen, way too much phosphorus, sulfur, magnesium, calcium, etc. And so all of that excess has to be dumped out into the soil in a soluble inorganic form that your plant can very easily take up. When you think about the form of nitrogen, for example, coming out of the bacteria or the fungi, because the same thing is happening there, the fungal feeding nematodes, the fungal feeding microarthropods, when they eat fungi, way too high a concentration for them to stay alive. And so they are going to poop out those excess nutrients. And of course, where is that going to be happening? Right in the root zone. Because here's where the highest concentration of bacteria and fungi are. So of course, the predators are going to come into the system eat the bacteria and fungi and release those nutrients in plant available forms. And so your plant just says, takes it, grabs it, pulls it into its body, ties it up on the sugars, and off they go to do their job of translocating into the different parts of the plant all the nutrients that that plant requires. So when we're thinking about nitrogen, for example, the form it's released, that is released from the predators eating the prey groups, the form is NH4, ammonium. When we're in a bacterial-dominated soil, the pH of the glues that these bacteria make are going to be alkaline. So typically in grasslands that are very productive, so in the soil where we have a lot of bacteria, we're going to have so much alkaline or high pH glue material being produced that it will cause that soil to be on the alkaline side. And alkaline conditions are what nitrifying bacteria need to take the NH4 that is present in high concentrations and convert that into NO3, nitrate. So early in succession, we'll talk a lot more about succession, not tomorrow, but the next day. So we want to have the right amount of nitrate if what you're trying to grow are early successional plants, but if later successional plants require the NH4. So it is the biology in your soils that control the chemistry, not the other way around. It's the organisms, well, and remember who's feeding them. Those organisms are being fed by the plant. And so what's the plant that is in the system? It's going to be trying to maintain that ratio of fungal biomass to bacterial biomass. So that plant will be highly productive, produce a lot of offspring. So do we need then the higher level organisms in this food web? Well, yeah, we do. Because if we get too many of these third trophic level organisms, they're going to overeat the bacteria and fungi, and all of the nutrient cycling is shut down. Yeah, your plant's going to be hurting bad. So we've got to have these higher level predators in the system to keep this trophic level at the right level. Not too many, not too few, so right at the right level. So the bacteria and fungi will constantly be eaten and released. Any excess nutrients that don't get taken up by your plant will be taken up by the bacteria and fungi that didn't get eaten by their predators. Do we need the higher level groups? Well, yeah, because we don't want this group, we don't want the fourth trophic level to get really high in numbers and overeat all of our third trophic level, which means nutrient cycling would shut down and your plant's in a world of hurt again. So we have to have the balance all the way through the system. Mother Nature is all about balance. It's very important to understand what's going on in these systems. So when you look at the organisms in the soil food web, with the bacteria, we separate the aerobic bacteria, the facultative anaerobes, and the true anaerobic species of bacteria. If we have aerobic conditions in the soil, if we're growing microorganisms, we want to maintain aerobic conditions. So this is the set of good guys. The facultative anaerobes, a huge number, probably the greatest number of our pathogenic or disease-causing bacteria occur in this group, the facultative anaerobes. They have to make enzymes that allow them to function in aerobic conditions as well as in anaerobic conditions. So they're having to produce two sets of enzymes, and therefore they cannot compete against the truly aerobic organisms that only have to make one set of enzymes in order to function. The anaerobic bacteria, of course, they need really low levels, less than 4 parts per million oxygen, whereas aerobic need above 6 parts per million oxygen. So we separate these good guys from the bad based on their response to oxygen. Actinobacteria are found very early in succession, so we want them there when you're trying to grow the brassica, the cole, the kale crops, the non-mycorrhizal plants, because many, not all, but many of the actinobacteria are suppressive. They will not allow the mycorrhizal fungal hyphae to get from the spore, once it germinates, to the root system of the plant. The actinobacteria may suppress the growth of the mycorrhizal fungi after they have colonized, and so your plant doesn't get the benefit. So we want the actinobacteria around at certain times, but certainly not if you're trying to grow a mycorrhizal plant. Well, different actinobacteria for different kinds of conditions and places and things like that too. So those are all factors that we have to consider. The filamentous fungi are aerobic. Mycorrhizal fungi are filamentous fungi, and they require the aerobic conditions, obligately aerobic, or their enzymes are not going to function and the organism's going to die. The yeasts, only a few of the yeasts are aerobic. All of the rest of them require reduced oxygen conditions. Omycetes have kind of been, their place in the taxonomy of organisms has kind of been changing over the last 10, 15 years. It's not really clear where omycetes are really going to end up. Are they going to be a whole new kingdom? They're not even going to be fungi anymore? Well, but they look exactly like fungi. They behave much like fungi. They have the same kinds of spores. And well, so for me, they're fungi. And sometimes, you know, you've got to differentiate between the splitters and the lumpers. Splitters want to give everything its own kingdom based on morphology and function. Whereas I tend to be a lumper where, hey, if they all look the same, if they all look like a duck, they smell like a duck, they quack like a duck, they're a duck. So that's my definition of omycetes. They tend to be very narrow diameter in soil. Now, take those hyphae of omycetes and put them on a petri dish with no competition and massive concentrations of food, and those omycetes become couch potatoes. Big, fat, blobby kind of hyphae. They don't look at all like a fungal organism once you put them into a plate count. But in the soil, where they don't have massive amounts of food and they're in competition all the time, yep, they're not going to be able to win under aerobic conditions. And so guess where most of the disease-causing fungi are found? In these two groups. A few in the ascomycetes, for example, in the deuteromycetes, yeah, there's some, but the majority. So I'm a lumper. Put together things that have similar function and similar appearance. So in the food web then, now we're into those things that eat the bacteria and fungi. The flagellates and amoebae, the ciliates are the bad guys. The nematodes, the good guys, the bacterial feeders, the fungal feeders, the predatory nematodes. Predatory nematodes eat other nematodes. Root-feeding nematodes, of course, the bane of growers everywhere. You don't want root-feeding nematodes in your soil. So how do we identify them? We teach people how to do these identifications. We're not going to take you down to the species level. We're not going to take you to the genus level. There doesn't seem to be a lot of reason to do that. So maybe I'm just, too wide-sighted to see what the identifying things to genus and species might have when we're trying to look at general function in the soil. Is your plant going to grow or not? Is a pretty generous question. So that's what we're looking for, is that kind of information. What's the fungal to bacterial biomass ratio, which will help us understand what kind of plant will grow? What are the constraints? Of course, microarthropods, we're in the process of adding these organisms into our soil food web picture. So when we go back to this picture, you'll notice that we've got one microarthropod that's representing a group of organisms that are, they're individuals. The individual species are in the hundreds of thousands, maybe even a million or more. How many species of bacteria are there? We don't know, because we haven't even begun to get around to identifying them all. Every time we do a DNA analysis, there's a whole bunch, maybe 50 or 60 species of something, but we only have those new sequences for them, so we don't know where they belong. We don't know, so this is a frontier, folks, that you're walking into, and a lot of unanswered questions have yet to be answered. So we're looking at this from a very general point of view. But who are the shredders? Who are the comminutors, the sapsuckers? When you think about an earthworm, they are comminutors. Earthworms do not digest organic matter. They are not decomposers. Nowhere in their genetic material are there DNA for coding for those enzymes that do decomposition. They chop up whatever they're chewing on, improving the surface area, the substrate available for bacteria and fungi to grow on, so you get a massive burst of growth because the bacteria and fungi have more food. It's not the earthworm that's doing that work. The earthworm did the chomp, 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 broke things down into small bites, which the bacteria and fungi now can just have a heyday growing on them. So we have to understand, well, I'm certain there are other things that microarthropods do as a community, working together with the other organisms that we have not even begun to suspect. We've got a lot of work left to do. It's the interactions of these organisms that form the higher level functions. Our overarching principles of what biology does in the soil are all based on the interactions of these organisms. If you don't have the fungi, you're going to miss a whole bunch of the overarching beneficial principles of what goes on in soil. So, the benefits of organisms, microorganisms, they are the ones doing all the work, and we've just talked about most of these. Nutrient cycling, we've talked about that. Retaining nutrients, bacteria and fungi. Suppressing disease, the whole food web working together will do that. Suppressing weeds. What we see, and part of the reason I related a little bit of this information about nitrate and ammonium, was as you have really high levels of bacteria, all turning all the NH4 into NO3, you are selecting for weeds. There's a recent set of scientific papers from Japan. I believe it's the University of Tokyo. Nakatani is the person who is leading the charge on writing those papers, has showed that the more nitrate you have in the soil, the more weeds you've got. As you reduce the amount of nitrate and increase the amount of ammonium, the weeds cannot grow, they will not be healthy, and your crop is going to win. Whatever your crop needs to be, you need to exit from a strictly bacterial dominated soil and turn that back into something with a lot of fungi in it. Just think though, every time we till, we slice and dice and crush the very organisms that you need to move things back into NH4. Every time you apply a pesticide and you kill most of the good guys in your soil, what are you selecting for? Weeds. We're doing it to ourselves because we don't understand what we're doing. Okay, so building soil structure. We have structure so water and oxygen and roots in your organisms will be held in an aerobic condition, and if you have that, there will be no erosion. You will not have runoff, no leaching. Everything's retained in that soil, so we stop destroying water quality of everything that's downstream of us. We have to stop pouring all those nutrients out into the ocean. That's not where we need those nutrients. We need those nutrients up here on the land. Stop letting them wash downstream. We're just crazy to be doing this. When we sequester carbon, putting all that elevated CO2 back into the soil from whence it came. And we can do that. Please go watch the animations on the Soil Food Web School website especially the one about sequestration of carbon, because we go through the math that talks about all of this. If we could all start composting correctly, it has to remain aerobic through the whole composting process. You've got to have all those nutrients and organisms present in that compost. So go through those animations and you can see the calculations that within 6 years, if everyone composted, all that elevated CO2 in the atmosphere would be back down into the soil and climate change would go away. Well, do we have the will to do that? Who are we battling? Who are we fighting? Yeah, follow the money and you'll realize why what we've been working on hasn't been paid attention to. So now that we're moving in that direction where most people have heard about this, people are starting to understand we need to be the rock that explains to people why soil life is important. How is it that we can get all that carbon sequestered back in the soil in such a short period of time? Maybe we should double the estimate. Maybe we should be saying, well, you know, given that we have to start up this whole industry and we've got to have composting operations all over, and a lot of the regs that are governing composting processes should be thrown out the window. The correct ones should be implemented. So lots of work there. And decomposing toxins. All those pesticides, all those toxic materials that we've been putting out into the soil for so long, we have to decompose them. Well, guess what organisms have been doing that for the last 3.5 billion years? Yeah, fungi. I have not found a toxic chemical that we cannot decompose. There is something somewhere out there that will deal with whatever toxic material you want to deal with. So let's get that decomposition going, so we don't have those problems in our fields. We're not having plants translocate those toxins into the food that our animals or that we as human beings are consuming. So when we think about what we do at the school, we do all the things that I've just been talking about. We identify the microbes. Please come and learn how to identify whether you got the good guys or whether you had the bad guys. We have to measure biomass or numbers of the organisms. We need to know about diversity. Do you have somebody working every second of every day, regardless if it's cold or if it's hot or somewhere in between, whether it's wet or it's dry or someplace in between, or if you've got this kind of toxin in the soil or you've got this kind of food? We need to have massive diversity in our soils. Where do you find that? Where do you locate it? And that's why you need to come and take the foundation courses. We want to select for the desired organisms and against the problem organisms. And I've already given you a big hint. You have to keep things aerobic. We teach people methods to enhance the desirable microorganisms in the soil and on your foliage. Oh yeah, there's the above ground part of the plant. And all of the organisms on the above ground part of the plant originally come from the soil. If you got good biology in your soil, you will have good biology on the above ground part of your plant, and you don't have the diseases, you don't have the pests, you don't have all of those problems. We want to help people, so we help people learn how to interpret the interaction. So with one microbe is high and the other one is low, what does that mean? If this is not quite right, but that is, what does that mean? And so we teach you how to make those interpretations. We want to enhance the beneficial organisms on the surfaces of food for our animals or for us. When you take a bite out of a carrot, you should want some of the soil. You should want some of the organic matter on that carrot, because that's going to be carrying the organisms that you require in your digestive system. How do we make certain that you are consuming on a daily basis those organisms that will replenish whatever sets of organisms got killed in your digestive system because you ate something with glyphosate on it. So we've got to get away from the vitamins. We've got to get away from taking a pill, popping a pill. You don't retain most of the vitamins or most of the minerals that you get. If you're popping vitamin pills, you're wasting your money. But you can see it yet another big part of the pharmaceutical world with a lot of money to try to convince people that vitamins and mineral pills are required and that they're worthwhile, when in fact they aren't. You have to have the organisms in your digestive system in order to be able to deal with what you put into it. So how do you get those organisms? You eat them on the food that you're consuming. So microscopes, yeah, we teach you how to use those microscopes. We're going to show you all kinds of different ways of assessing the biology. This happens to be the surface of some compost, and you can't even see the surface of the organic material in here because it is so covered with all these organisms. We've got lots of bacteria in here. We've got all these strands of fungal hyphae, and you can see how some of them are really wide and some of them are very, very narrow. And these are yeasts, and just herds of different species, different sizes and shapes. And that's what we want to be seeing. This is a good compost. When we're making compost, you need to understand the specific recipe that's needed. You have to make certain the biology is growing. How do you measure that? You need your microscope. This is a soil that's been improved by adding biocomplete compost. And look at all the species of bacteria. Look at all the airways and passageways for oxygen and water and the roots of your plants to get into these cracks and crevices. Good nutrient cycling. Take a look at the anaerobic version. This was in the plot right next door. Horribly compacted. No air, well, maybe one or two, but that's it. Airways to get oxygen and water and organisms in your roots. Yeah, you are not growing good plants here. They're going to be sick, unhappy, unhealthy, and they're going to be diseased. And you as a grower, not going to manage. So we make liquid versions of these organisms. So typical tea brewers, extractors, simple. We don't want anything inside. We want an air pump blowing into here. We want to put the compost in the bag and immerse it in the water. And in 30 minutes, you'll have your extract, or in 24 to 48 hours, you'll have the tea where you've actually brewed more organisms. And then again, a representation of the human digestive system. If you've got poor sets of microorganisms in the soil, that means you're not going to have good plant growth, that's not going to be healthy, you're not going to get the nutrition that you require. In conventional agriculture, we continue to kill, destroy those actual sets of organisms that we need, so they aren't present, and that's what promotes poor growth of the plant. Get those organisms back into your soil so you can eat the organisms that you require and get them back into your digestive system, and your food will appreciate it as well, as will you, as will your digestive system, because you'll be consuming the organisms, the good organisms, on the surface of whatever you eat. So hopefully that's an exciting beginning to the Soil Summit, and I hope that all of you enjoy today's speakers.
So that was the famous Elaine Ningham. Amazing, amazing stuff. I have a little bit of an exercise for you to do. I want everybody to choose 2 or 3 words about what you've just learned, the words that stick out for you, the big words that might seem important or you might have some questions, and just throw them into the chat. Let's see how that landed with everybody here. I'm going to go with some that are already there, but as you're doing that, Vinay has said, so what is the correct amount of tilling one should do? you should do very little tilling. You should do ripping. The problem with tilling is churning the soil. It is destroying the microbial community. It is destroying the structure of the soil. It's doing everything that we don't want to happen. And to do tilling, which a lot of people do with tractors and things, it requires that there's also nothing on the land, or results in nothing being on the land. It brings the soil up, it dries the soil, it dries the microbes, and it emits carbon dioxide. If you remember the very first video that we saw when we looked at the NASA video of carbon dioxide in the atmosphere, it was during that time of plowing and tilling. Tilling and plowing, there's not that much of a difference. One is just slightly shallower than the other, but it's still disrupting the soil and emitting carbon dioxide. A lot of that carbon dioxide that's being emitted are microbes that are dying, okay? Soils that are drying. So if you've got very compact soil, you just want to rip it, just make some deep lines in it, get some extract in there of your microbial biocomplete compost or vermicompost or extracts or whatever those are that you've got, and get your roots in as quickly as possible to keep those microbes alive through photosynthesis. So what's coming in? What do we have? How does one distinguish between good and bad nematodes in the soil? If you don't have a microscope and you are not trained in looking at the bad nematodes, you'll see it in the damage that is being done. The root-feeding nematodes will damage the roots of your plants. But back to the functioning groups of nematodes, there are predatory nematodes that will consume the root-feeding nematodes. The only reason those root-feeding nematodes are there in abundance is because you're likely to have an imbalance. There's nothing to predate on them. If we had 100 lions outside my window here, every single thing that a lion would eat would get eaten. We need to have, well, I don't know what predates on lions, actually. That's a really bad one. Lions are the kings of the jungle. Man, we need to have hunters. That's so bad. But you know what I'm saying. It's all about balance. Okay, when we've got balance, we've got nutrient cycling, we've got nutrient retention, we've got soil building, we've got less runoff, less erosion, less compaction, we've got sequestration of carbon, and we've got decomposing toxins. Photosynthesis, Wesley says, yep, absolutely important. Vivian, aerobic, symbiotic, balance, fantastic. Mwenga has said filamentous fungi, super. Duncan, independence, great. Wesley, benefits of microorganism, absolutely yes. You're beginning to see the biology drives the chemistry. Barbara, earthworms are comminuters, not communicators, comminuters. They break down things into smaller bites, and bacteria and fungi are able to get a lot more food due to this. They work together. The entire community is what makes it work. J, microarthropods, exactly. And remember, we're talking about massive species differentiation, thousands, if not millions of different species of these organisms all working together. In the video that I was taking yesterday of the water infiltration, you saw me zoom up and look at the top of the second sample that I was doing, the infiltration. You saw all those microarthropods in there. There were ants, there were tiny weeny spiders. I mean, it was crawling. It was literally crawling. There was a whole bunch of stuff up there. So important. Carbon sequestration, Florence, absolutely. Composting will reverse climate change. Looking after our soil biology is going to reverse climate change. Yes, composting is one way of getting us there. How often should we rip or work on the soil? You should only really need to rip the soil once. If you've got compact soil, you're trying to get rid of the compaction. Why is the soil compact? If the idea is you rip it all the time and you rip it all the time, you're continually disturbing it. The microbes will do that. The plants, the roots, the way you plant, the way you grow will do that work, okay? The idea is not a ripping and tilling and plowing are a man-made construct for agriculture. In the absence of understanding and even knowing and even seeing and even having microscopes or the cutting-edge science that we have today, even understanding that soils function without being ripped. They function without being plowed. They functioned before we were there. We've put those processes in place. And in the process of doing so, let's go back to the first video I shared. within 30 years, we've destroyed, 100 years, we've destroyed 30% of our topsoil. Okay. Interactions of these organisms is critical for great soil, absolutely. Biodiversity results in both micro and macronutrients. Yes, absolutely. And that nutrient cycling is all dependent on these guys. Vivian has said, do you agree with Dr. Ingham that if people compost correctly within 6 years, all excess carbon can be sequestered in the soil? You know, there are millions of farmers across the world destroying soils as we speak, small scale and large scale. If, as she says, everybody changed, I believe yes. Because we would begin to, you saw the NASA footage. What happens when the plants are pulling down the carbon? That was 1 year of footage. And that was just a season, and we were seeing basically the impact of what was happening in the Northern Hemisphere with their planting programs, not so much the Southern Hemisphere. It is amazing how soils and ecosystems can bounce back, literally, if we leave them alone and work with them. Have we got any comments from the YouTubers? Malawi and James and Shiro. Hey guys, anything, any comments? What are the big words that came out of this for you? Now, I'm going to put a video into your WhatsApp group. And James, you're going to remind me, the video by Andre Loy. That question of, do we believe? And he talks about what can be done across different production systems to sequester carbon in certain amounts of time. Towards the middle, towards the end of the video. Really, really powerful stuff. So please do watch that, okay? The other thing that we're going to do, James, is we're going to put in the scholarship link into this group for any of you that want to pursue and follow the Soil Food Web program. Fill in their scholarship link and apply. If you're lucky, you'll get a scholarship. And if you're not, apply again till you do, okay? I'm going to take you now on to the next part of today's lesson. And what we're looking at is, we've looked at plants bringing down sugars, feeding them in, and the nutrient cycling that's happening. There was something that Elaine said earlier on, and I wrote it down here, and she said, basically, when we look at a system like that, we can look at it by following the nutrients through the biology. We are part of the system. We are another trophic level somewhere out there as well, because of how we interact with plants and microbes, etc., etc. Following the nutrients, and this is where we're going to be going also with this course, is how do we inoculate our soils, but also what is the pathway of the nutrients? Because as I've said from the beginning, we're not growing food for the sake of growing food. We often make that mistake. We have, Ministry of Agriculture and Ministry of Health. They're so intrinsically linked because health is dependent on food. So if we grow food badly, we end up with sick people. The next presentation is going to just talk a little bit about that for you to understand a little bit about the nutrients, the things we're calling insects, and bricks. So get your notes out, your notebooks out, or your pen and notebook out, and get ready. And any questions that you've got, just be throwing them into the chat there, any observations as we go along. I do have a couple of exercises for you in this presentation. So let's go. So this presentation is called Soil Microbes and Bricks. And we will come to what bricks means. Simplifying what we've done so far and what we know. Photosynthesis, energy from the sun, carbon dioxide, water, plant roots, healthy plants grow. They do need micronutrients in a healthy system. They get the micronutrients from a healthy soil microbiome. We've just been told that by Elaine. We've been shown how it works a little bit by Christine Jones, and plants grow and they're healthy. That's the norm, right? The liquid carbon pathway, pumping that carbon in, feeding those microbes, plants, everything being healthy, okay? But this is what we've done with conventional agriculture. We've ignored that there was even a soil microbiome, that it even exists. We've said, oh, we need to put chemicals into the soil, micronutrients into the soil, and literally put them in synthetically, give them to the plants. The plants then do not need to spend that much energy communicating with the soil microbiome. So the biome dies because they're not getting fed, because the plants are sucking up through osmosis these very soluble and easily accessible nutrients, because they're salts. The plants also can suffer from toxic stress or other things by these salts. And the reality is 80% of the fertilizers that we put in the ground leach out. The plant can't absorb them all. So when it rains, they leach away and they get into our waterways, grow our algae blooms, etc. But the plants get a bit lazier, ground becomes compact, you get erosion, soil erosion, you get compaction, you get pests and disease, but we can fix that, guys. We just bring in toxins, right? That is a typical scenario of the way we grow conventionally. All of those inputs, those 2 syringes, that's big pharma. You got to pay for that and pay for that. Okay, but we created the problem ourselves because we didn't understand that the chemistry is driven by the biology, not the chemistry is driven by an agro dealer or big pharma. Okay, let's look at bricks. So what is bricks? It's a measurement of the dissolved solids in a plant's sap, which indicate how well it's photosynthesizing. Now, isn't it so cool that we have an instrument that we can actually squeeze the sap of a plant and look at how well that plant is photosynthesizing? Okay, a bricks reading is normally for plants between 1 to 25, sometimes higher 30. If you're using it for management purposes or for nutrient purposes, remember it's dissolved solids, and those solids include those nutrients that are cycling in and out of that plant. So this is what it can indicate: nutrition and taste, measure of the sugar content in fruit or vegetables. Higher bricks is related to higher sugar value and better taste. Plant health, a higher bricks level indicates a healthier plant that's more resistant to pest. And we're going to get into how insects, what that relationship is between pests and bricks. Photosynthesis, a higher level bricks indicates that the plant is photosynthesizing well. If it's photosynthesizing well, we know that it's communicating with the microbiome well. Turgid pressure, that's a high level bricks, indicates that the plant has better turgid pressure of its cells. This gives a plant firm leaves and makes a plant also resistant to diseases and pests. Plant has got more vitality, more immunity. Nutrient deficiency is a lower bricks, may indicate that the plant is deficient in certain nutrients. So this is a refractometer. It's a tool to measure bricks. The one on the left is like a little telescope. It's a manual one. The one on the right is a automatic one. A digital one. You put a drop of sap from your plant leaves typically onto the instrument, and then you take a reading. You squeeze the sap out. You can use this is a garlic press. You can use different ways to actually get the sap out, squeeze it, put it onto the little reading thing there, and you'll get a reading, okay? So let's look at bricks again. We said that bricks is a reading that can go from 1 to 25 to 30. Plants that have a bricks reading of 1 are typically not very healthy. You do not even want to be eating a plant that has got a bricks of 1 yourself. Anything below 6, the second one there, here we go. At about 6, plants are still not that healthy. And unhealthy plants have got thin cell walls. They attract pests, different bricks kind of readings. You can actually tell by the pests that are on your plants how healthy your plants are. So what we're learning now is that in the absence of instruments, in the absence of a microscope or in the absence of a refractometer, you can still read the indicators on your plants through insects. We've learned that you can read the indicators through weeds. A lot of weeds indicates a lot of nitrates in the soils and no ammonium. When your plants start, when your soils have got a better nutrient cycling and there's more ammonium in there, you'll find less weeds. Those are things if you don't have instruments. You do not need high-powered instruments to do this. You need knowledge and curiosity, and you need to be observant, okay? So there we go again. After about 12, plants grow well. They can protect themselves from pests and diseases once they've gone past that Brix level of 12. We want to be eating plants that are on that side of the spectrum, okay? So we say that photosynthesis and the soil biome drive Brix. Brix changes throughout the day because photosynthesis changes throughout the day, okay? The sun moves from morning to evening. Plants will use their little solar panels, their leaves, to get sun. We all know that at midday it's the hottest. In the morning, different rays as well as in the evening, but that difference can also be blocked by clouds. So the entire process throughout the day is different, but healthy plants typically will have a difference of 3 Brix points from sunrise to sunset. So if you're using Brix in the Brix refractometer as a tool to see how healthy your plants are, they may look healthy, but you can actually judge how nutritionally healthy they are. Then you would want to take a reading in the morning and a reading in the evening, and you'd expect to see about a 3-point difference. And the difference is because just at the end of the day, the plant, when the sun goes down, will start really pumping those exudates down into the microbiome, the rhizosphere, the rhizosheaths. And in the morning, there's less in the leaves when you do the test in the morning. Now let's look at bugs and Brix, okay? Here's a leaf Brix chart. And if you look at the picture of the leaf, it's got 1, 2, 3 on one side all the way up to 16 plus. At 1, 2, 3, basically, nobody wants to be eating that, not really even insects. Or insects do, sorry, not higher insects though. The aphid groups and the sap sucking insects will come in around 4 to 6, and you can see there's 6 to 8 on the Brix. Sucking insects 7 to 9. If you read the part about the insects, it's about their mouth parts. When a plant is weaker and the cell walls are weak, the aphid groups and the sucking insects find it much easier to get into the cells. So if you're looking at your plants and you can see you've got aphids on them, you know that that's the problem. It's not that the insects are damaging your plants and that's why your plants look weak. It's that your plants are weak and they are attracting insects that can consume them as weak. We're taught the opposite. We're taught that if you just take an insecticide and spray the insects off, your plants are better. They're not. They're now plants with toxins on them. And those toxins will keep the insects off, but the plant nutritionally is not well. Yeah. If you look between 9 to 12, you get chewing insects. After 12, we've talked about the grasshopper group. After 12, sorry, you also end up where there's very little insect and very little disease because a plant can defend itself. And that is where we want to be eating. We want to be consuming food with a higher Brix than 12. We're not insects. If we're eating food down in the 6 and below, or the 9 and below even, it's not that the insects are competing with our food. We're competing with insect food because it's not even nutritionally valuable food for us. Okay? So we want our plants to have a high Brix. This method of agriculture, which is using synthetic inputs, driving plants that end up with pests and diseases and then just solving those with chemical inputs is because the plants have got low bricks, because the plants do not have a functioning microbiome, soil microbiome. So let's look at bricks and us. Here is a recent experiment we did. We said, okay, so what do we get on the market? When you go shopping to buy food, what are you buying? And why are you buying vegetables? What choices are you making when you're buying your vegetables? I personally feel that we're buying nutrients, right? We're not saying, we're just going to buy anything, fill our belly. We're trying to grow ourselves, nourish ourselves, feed our families with as nutritious food as possible. But is the stuff that ends up on the market that? Is it nutritious? Is it toxin-free? Is it safe? Is it of the value of the money we're paying? What choices do we make when we go there? And when we think about where we go and buy our food, and we think about illness in livestock and others in feedlot systems, and we begin to think, well, actually, the human feedlot system has become the supermarket because we go there, we're presented with things, and we trust this is nutritious and safe food. But is it? This experiment was done with 3 different types of leafy greens, kale, spinach, and managu, which is one of our indigenous varieties here in Kenya. We purchased on that morning, same morning, I had somebody go and collect from 4 different sales points. The first one is a large supermarket, Carrefour. Second one is like a boutique kind of shop, supermarket, care and provision stores. Then we bought some stuff from Mama Mboga, which is like your side road kiosk, and then a mid-range supermarket, which is Cuba. Okay. If you look at those now, which would you choose and why? If you've got your pen and paper down, make a choice. Where would you buy? If you had a choice of going to those different places, or if you had a choice of just going to one, which place would you choose to buy your food and why? So you can start actually throwing that information also into the chat. Let's have a look and see what you're saying on chat. And let's have an idea of why we're choosing the things we're choosing. I'm going to give you a little bit more information as well. Okay. Now I've overlaid the image with the weight, and also with the price, and also with the number of stems. Now that also might influence your buying choices. Take a look at it for a minute, and let's have you put down your buying choices. Okay, so put your buying choices there in the chat. Vinay has said he will buy from KPS. Do you want to write in why? Charles, you said you'd buy from Mama Mboga. Let's write in why we would make those choices. I can see that Elijah has said Carrefour looks healthy. Peter Oswata has said from Mama Mboga. Anybody else going to put some choices in there? You guys on YouTube, do you want to throw in what you would buy and why? Lawrence has said the kiosk stuff looks healthy. Vinay is buying from KPS because they will have some form of a supply chain and traceability. They will have. They didn't say it on the packaging or on the labeling. So again, it's an assumption. We assume that they will have. Okay. Neighborhood mama who I know. We're talking about exactly what you're seeing in the picture now, not not anything that you're not seeing, not what you hope or what you do. But if you had a choice of what you're seeing in the pictures, yeah. And for some of you, you might prefer to see these things graphically. So there we are, we've got some graphs that show us the same thing about the price, which place had the different prices, which place had, the first one's price, the second one is weight. That might help you. So James Muya has also said KPS. Okay, well, we'll move on and you can keep writing these things in as you make your minds up. So moving on, Mamamboga, Nathan has said Mamamboga. Well, okay, now I'm going to overlay them with their bricks, with everything that you have learned about bricks, okay? So there are the bricks, figures of these things. Question is, which one would you choose to buy now and why? And would you change your mind? Remember what we've learned? That bricks above 12 is what we should be looking for. That is human food. Everything that is below 12 is likely to actually have some kind of pesticide on it if it got to the shop looking healthy and nice. Okay, not necessarily looking healthy, but like it didn't have those insects on them. Okay, exactly that, Barbara, your point there. I'm going to go shopping with my refractometer. One of our guest speakers who will be joining us at the end of the course is building a tool so that consumers can make the right choices based on nutrients, because we're not getting the nutrients that we should. We are seeing illness in humans. We are completely blind as to what we're eating when it comes to safety and the toxin levels, okay? I can see more people now have turned to Mamamboga, but guess what, guys? This was only on that day. Tomorrow, Mamamboga might be supplied by the same person who is supplying Carrefour. And Carrefour probably has and all the others and all the big supermarkets probably does have a traceability system in place and know your suppliers, but do they have a track on the nutrients that are coming from those products? Probably not, because they just want big leafy green things, and you can get that if you're using synthetic chemicals. They can look big and leafy and green. They're not very nutritious. And to protect them, because they're not nutritious, from pests and diseases, we spray them with a lot of toxins. So my point, and I hope I got that point across, is we don't really know what we're buying, when we're buying. Bugs help us as an indicator. Okay, when we look at this here, we look at the bug chart, we knew that everything below 8 was aphid food. Carrefour was, at that point in time, on that day, had products that were good for aphids, would have been recognized by aphids as being their kind of food. We, unfortunately, don't even know how to recognize what is our kind of food. Then we looked at sucking insects, we've overlaid there, the sucking insect part, we've overlaid there the chewing insects, then we've got to the 12, which is the cutoff point, and we want stuff above 12. And there isn't a lot that was above 12. Okay. Mamamboga did come out. But if we all went shopping that day in the region where these things were purchased for our vegetables, we wouldn't have been buying real nutrients. We would have been filling our bellies with things unlikely, with toxins as well. So just coming back to this here, we're saying that soil microbes. I've got something on top of my screen here, which is just covering what I'm covering my screen. Let me just try and get rid of this. Sorry, 1 second. Can't get rid of it. But anyway, the big takeaway is that soil microbes drive nutrient density and thus the health of plants, animals, and us. So I'm going to exit that, stop sharing that now from there. And we have a few minutes to talk about that. Who wants to talk about it? So Barbara, I know, is like online looking, where am I going to buy my refractometer from? And remember this, that the reading you will take can be influenced by the time the product was picked. If it was picked in the morning, if it was picked in the evening, but you can look for other indicators. If I used to say, if it's good enough for bugs, it's good enough for me. And now I say, if I see chewing insects have been eating it and I have to buy it because I didn't grow it by myself, I would rather choose something that had the damage of chewing insects than the damage of sucking insects. But most time, I would grow my own or buy from somebody who I trusted and I knew and who was producing regeneratively. Nice to see that Barbara already has a refractometer. Great. I can just see her in the store there being sent to the manager's office. What are you doing? And she's squeezing all the juice out of these things in the aisles. Barbara, don't get yourself in trouble. But if you can, go and show your supermarkets, because we've got to push we have to push for safe foods every which way we can. And if part of that is sitting down in a supermarket and showing those guys what this means, yeah, then there we go. James says, is there a way we can know the healthy produce without the gadget? As I've said, look for symptoms of chewing insects rather than sucking insects. So the sucking insects leave tiny little holes, little pinpricks, or you see aphids. Chewing insects will actually take out bits and pieces of the leaves. But just know that if you're buying something and there's no insect damage on it, chances are it's sprayed out, because this stuff is food. It's food to us. It's food to the ecosystems that are around it. And there will hardly be a time where something will be produced and there won't be some kind of predation on it. Let's not call them pests, predation, something that will want to eat it. Yeah. So we've got a couple of minutes left. I'll take 2 speakers from the floor. Does anybody want to put their hand up? And Mama Mboga wins in this scenario now, but remember this, it changes every day, because supply chains procure from different places. Often that changes as well, whether it's a Mama Mboga or a supermarket. Any hands up? Who's brave? Who has something to say? Or are you all just Googling refractometers and just saying, wow, 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 what are we eating? What are we feeding our children? What is going on? Vinay. Let's give Vinay the microscope. There you go, here's a microscope. The microphone. Vinay, you'll have to unmute yourself. Put your camera on. Go ahead.
Hi. Hi, Sue.
Hi.
Hi, everyone else. Okay, my thing is, okay, in mangoes, we do a lot of mangoes at the farm. I've noticed the bricks value, they change around our irrigation pattern. I don't know how that explains, because once we irrigate towards the end of we irrigate once a week, the mangoes. If straight after irrigation, if we harvest, the bricks value are low, but towards the end of the week before the next irrigation, the bricks value go higher.
What are you checking the bricks on?
The refractometer,
Yeah, but are you doing the fruit or the leaves?
the fruit.
Okay, so don't be doing the fruits are indicating the sugar levels, and the sugar levels, when there's less irrigation, the sugar levels are going to be higher in the fruit because of more concentration of those sugars. So that is to be expected. So when you are irrigating, there's more dilution of those sugars, you'll find that the bricks will be lower. And that's why when it gets very, very dry, fruit becomes very sweet. Yeah, those sugars are more concentrated. But to do it from a management perspective, to know what's happening at the roots, do the leaves. Do the young leaves and take your bricks readings in the mornings and the afternoons with your leaves, and they will tell you if the biome is functioning. And what you're getting, the difference between the morning and the evening should be about 3 points. Yeah. Do you have any problems with the mangoes? Are you getting diseases? Are you getting pests? Are you getting issues that you should be concerned about?
Pests would be mostly fruit flies and seed weevils.
Okay, are they, is it a big problem?
You have to take care with pheromones, otherwise, yes. So is it naturally the plant can repel fruit flies and the seed weevils because
All plants should be able to defend themselves. So if you've got what is an infestation that you're having to deal with, then you could probably do with improving the soil biome around the roots, that make sure just that the soil biome is working properly, and the plants should be able to defend themselves. So that also looks at what is growing around the plants, what's on the ground around the plants. Is it bare? Is it just mulched? Is there any nutrient cycling happening?
yeah, we have started on the regenerative path, but my question is something like a seed weevil. It's not infestation immediately. It just lays an egg in the flower. And then when the flower grows into a fruit, it is inside the seed. The weevil will grow over the 100 days, next 100 days.
Yeah, I have to say, I don't know about insects that are going into flowers. I would imagine, what are the predators of those weevils?
None that we know.
I would advise you to look online and see who the predators are, because there must be things that predate on their eggs. There must be things that will predate on the adult bugs. And if you don't have an ecosystem with those predators in there, then that's what you need to be trying to put in there into the system.
Okay, that's good. And one last quick one, again, going back to the bricks.
Mm-hmm.
So the fruit bricks value can be deceiving towards your plant health or plant bricks or plant nutrition uptake, isn't it? Because it
Sorry,
cannot give you, if you measure your fruit, you will not have an accurate status of your plant health.
you're just getting a measure of more likely yes, no. Sorry, yes, no. You're more likely getting a measure just of your fruit sugars. You're not getting the nutrients in the nutrient cycling in your plant. The fruits have already done their, that's the end part. You want to do it with leaves, because you're doing it with the photosynthesizing parts of the plant.
Okay, that's clear. Thank you, sir.
Perfect, cool. So any other question? I don't see any hand up, guys. We're 3 minutes over time, so we're going to do what we know. Hesbon and James, take me off. Put us all into the gallery, give us all microphones, give us all cameras, and we can all say good night to each other. And Sylvester Kilunga, I can see that you chose Mama Mboga. Good. I hope you were doing it after you saw her bricks. Goodbye to everybody on YouTube, and good night and goodbye to everybody here. Don't forget to do your homework. Looking forward to those wonderful videos. You can show us with a thumbs up if you've enjoyed today's class. And bye-bye, and we'll see you all next week.
Bye bye.
Bye.
Thank you so much. Bye.
Thanks, Sue.
Okay.
Bye.
Thank you.
Bye.
Thank you.
Bye, everyone.
Bye. Bye. Bye.
Bye-bye,
Bye,
everyone.
everyone. Yes. Bye. Bye.
Yeah, I'm somewhere to bye.
Bye.
Bye. Bye.